Dear listeners, welcome to another episode of the podcast about enzymes and molecules. I am Roberta Foresti, Professor of Biochemistry at the University of Paris-Créteil and researcher at the Mondor Institute of Biomedical Research in France. For our fourth episode, we will talk about a special system that is very important for protecting the organism against stress, the NRF2 transcription factor. It is the master regulator of the stress response, which is responsible for increasing the expression of a long list of genes that help to combat stress and restore cellular homeostasis. Interestingly, NRF2 is activated by stressful stimuli but also by plant-derived natural compounds, showing how nature has evolved to harness the power of plants to provide benefits for our body. In opposition to its fundamental role in protection, NRF2 also has a dark side in the context of cancer, since its overactivation in cancerous cells renders them stronger and more resistant to the drugs that aim to kill them. Today we welcome a researcher that has greatly contributed to understanding the detailed mechanisms underlying NRF2 activation by different stimuli and is actively engaged in examining the role of NRF2 in metabolism and cancer. I am delighted to host Dr Albena Dinkova-Kostova, originally from Bulgaria and currently Professor of Chemical Biology at the University of Dundee, Scotland. She obtained her PhD in biochemistry from Washington State University in the USA and joined the laboratory of Paul Talalay at Johns Hopkins University, where she became interested in chemoprevention by chemical and dietary induction of cytoprotective proteins. After several years in the USA, in 2007, Dr. Dinkova-Kostova joined the Medical Research Institute at the University of Dundee as a Research Council's UK Academic Fellow. Her work is focused on the mechanism of induction of cytoprotective proteins through the KEAP1 NRF2 pathway and the chemistry of inducers. She has an impressive record of publications and has also been nominated on the global highly cited researcher list for 5 straight years from 2019 to 2023. Hello, Albena. Thank you for your availability. It is a pleasure to have you today as a guest for our podcast.
Hello, Roberta. Thank you very much for inviting me and giving me the opportunity to contribute to your podcast, which I've enjoyed all of them so far.
Thank you. Before we start with the questions, I need to thank somebody who has been helping me a lot for the preparation of this podcast. And I already told you before in our previous conversation, he is my son, Matteo Motterlini. He is a data scientist working in a company. He doesn't do any research or science, but he helps me a lot with the editing and the preparation of the videos for this podcast. So I think I will add a picture of him in the next, in our next episode. And when it is published, he's going to get some recognition for all the work that he does to help me.
That's great. Well deserved.
Yes. So to kick off, Albena, could you give us a definition of NRF2 and how it is activated?
Yes, of course. As you mentioned already, NRF2 is a transcription factor that regulates the expression of many genes, probably more than 200, depending on the context. The products of these genes are proteins with very diverse protective roles. At homeostatic conditions, NRF2 is a very short-lived protein, between 10 and 20 minutes half-life, because it is subjected to continuous degradation. And one major contributor to this degradation is a protein called KEAP1. KEAP1 binds NRF2 and brings it to a complex that promotes the ubiquitination of NRF2 and its subsequent degradation via the proteasome. When specific small molecules, which we call inducers, enter our cells, they bind to KEAP1 and prevent its ability to target NRF2 for degradation. This leads to NRF2 accumulation, translocation to the nucleus, and upregulation of its downstream target genes. The protein products of these genes have various protective functions. They ensure, as you already mentioned, Roberta, adaptation and survival under various conditions of stress that our cells encounter. And they act to restore the normal homeostasis.
NRF2 is continuously produced and is continuously degraded. So this is a signal that the cell is trying to be ready, right, for a kind of possible potential stress that may be coming. We don't know when it is coming, and the cell is ready to react in case of whatever stressful stimuli it is coming. This is how we can interpret the fact that NRF2 is continuously produced and at the same time continuously degraded if it's not needed, correct?
Yes, that is exactly correct. In the beginning, we were wondering why should a cell invest so much energy in making something only to destroy it as soon as it's made. But as you precisely defined, this is because we need its availability immediately under conditions of stress. If the cell has to rely on a different type of signaling, for example, which involves de novo gene expression of NRF2, that will take many hours. And if the insult is severe enough, the cell may die in the process. So this is why we think the cell undergoes through this effort to make NRF2 continuously and destroy it.
Yes.
But ensuring at the same time that it could be stabilized as soon as it is needed.
Yes. So it means that this energy spent for, as you say, the effort to make it, it means it's really, let's say, another way of recognizing the fundamental role of NRF2 in the cell. I think this is how I view it.
Yes, definitely.
So I wanted to continue by talking a little bit more about KEAP1, the repressor of NRF2. I read that there is this cysteine code, which is very important for activation of NRF2, and that the cysteines on KEAP1 have a particular role that you are going to describe to us.
Yes, sure. KEAP1 is indeed a protein that is rich in cysteines. And cysteine is a very special amino acid because it has the ability to undergo oxidation to various oxidation states, some of them reversible, others irreversible. In addition, cysteine can form covalent bonds with electrophiles. Electrophiles are molecules that can bind to nucleophiles, and cysteine is one example of a nucleophile. KEAP1 is particularly special in that it has cysteines that are highly reactive due to their local environment within the protein structure. Some of the cysteines of KEAP1, for example, are surrounded by basic amino acids, and that increases their reactivity so that they exist as thiolate anions ready to bind electrophiles, even at neutral pH. When we say cysteine coat, we mean that inducers, or these NRF2 activators, preferentially bind to certain cysteines to KEAP1 and not randomly to any cysteine. We also refer to these cysteines as sensor cysteines, as they are the ones that recognize the electrophilic NRF2 activators and trigger the induction of these cytoprotective proteins. I think the idea and the term cysteine coat was originally invented in Masayuki Yamamoto's lab when they tried to classify different inducers, the preference of each inducer to specific cysteines that we know are particularly reactive in KEAP1. What we still do not understand is whether modifications of any of the sensor cysteines in KEAP1 leads to the same transcriptional response, or whether the transcriptional response is more nuanced, depending on the precise cysteines that have been modified. So this is still an open area of potential discovery by the younger scientists.
I see. So I was thinking in a different way, but you tell me that it's a little bit more complex than what I was thinking. I was again referring to the thinking that certain cysteines are sensitive to certain inducers was a kind of specificity. Certain inducers that are produced during particular stress situations will act on some cysteines instead of others. But you're saying that it's more complex than that. It's really also what inducers will attack with cysteine and which genes may be affected.
Yes,
particular reaction.
Yes, you're absolutely right. This is the idea of the cysteine column, this specificity of specific cysteines reacting with certain inducers. But, so this is relatively well understood based on many mass spec analysis and also, crucially, mutagenesis analysis in cells and even in animals. But the reason that I mentioned that what we don't understand, this is the aspect, the last aspect, the more perhaps nuanced possibility. It may or may not exist, but it's something that we can be open to, whether the specific transcriptional responses may also depend on a certain type of cysteine that is modified. We just don't know that yet.
Very interesting. So, different inducers, although they will all induce NRF2 accumulation in the nucleus, may lead to different transcriptional regulations. Is that what you are saying?
I wouldn't exclude that as a possibility.
Interesting.
We don't know anything about it, but it is not impossible. And I think mainly some of this possibility could be because some cysteine acting molecules, they modify the cysteines of KEAP1 reversibly, and some may modify them irreversibly. Particularly, we don't really understand very well how oxidants can do that. For example, hydrogen peroxide reacts with very specific cysteines, which are completely different from, let's say, bardoxolone or mevaloxolone, the triterpenoids. And we don't really understand. There will be definitely overlapping targets, but it is just possible that if the reversal of those cysteines to their normal state has different kinetics, maybe that would also determine the duration of NRF2 accumulation, and that may be reflected in the downstream target gene expression. We just don't know. We don't understand it yet.
I see. So looking a little bit more into these chemical structures of the inducers, which seems to be very important. We know that NRF2 inducers have to have some particular chemical features in their structure that are required for them to be able to induce NRF2. And you have worked extensively on this. What are these features, and why is their presence so important for their NRF2 activating properties?
Yes, so far we can group the NRF2 inducers in two large classes. One class of compounds, which is the younger class, more recently discovered, are compounds that bind to KEAP1 non-covalently. And by binding to KEAP1, they are specifically designed to bind to the couch domain, which is the domain of KEAP1 where it interacts with NRF2. And that binding prevents the interaction between KEAP1 and NRF2. We call them protein-protein interaction inhibitors. The class that we know much more about and which was discovered even before NRF2 are the electrophiles that I already mentioned. Electrophiles bind covalently to specific cysteines in KEAP1, as we alluded to in the previous question, and they disable the function of KEAP1 to target NRF2 for degradation. In fact, in the 1980s, a decade before the discovery of NRF2 and KEAP1, electrophilicity was recognized as the only common property among compounds that induce genes that we now know are regulated by NRF2. For example, these are genes encoding quinone reductase 1 and Q1 or glutathione transferases. And this inducibility by many electrophiles was found to protect against toxicity and carcinogenicity in models, rodent models, using rats and mice. And the recognition of the electrophilicity as the only common feature among a diverse array of compounds that can induce the gene expression of these particular two enzymes led really Paul Talalay and his colleagues at Johns Hopkins University at the time to predict the existence of a sensor protein with reactive cysteines. This idea was not welcomed very readily in the field at that time because it was a ligand-receptor interaction era, and everyone was looking for some common structural features among small molecules that affect the function of a protein. But this was not the case from their study. So because electrophilicity was the only common chemical signature, they proposed the existence of this sensor protein that has reactive cysteines. And this protein was shown to be KEAP1 by Masayuki Yamamoto and his colleagues in Tsukuba University in Japan. And most of the electrophiles that were studied, both in the Talalay lab as well as in the Yamamoto lab, at the time were exogenous. They are present in our environment or our diet. But more recently, it's very interesting that various investigators have observed that certain endogenous electrophilic molecules can also bind to KEAP1, causing NRF2 activation.
Interesting. So this electrophilicity is a key feature which is found in exogenous substances that you basically started with these substances. But now we are discovering that also there are many endogenous metabolites that do possess the same property, chemical property, which is not structural but is chemical. Yes, and they are produced in the cells under stress conditions, okay, and they will induce NRF2. Can you then give us some examples of these molecules and how they will connect metabolism to the stress response? Because they are metabolized, they are produced during particular conditions, and as a consequence, they will activate the stress response.
Yes, exactly. So electrophiles can be produced in our cells during certain physiological and pathological processes. For example, probably some of the earliest recognized electrophiles that were shown to activate NRF2 are lipid peroxidation products, such as 4-hydroxynonenal or the cyclopentanone prostaglandins. And both of these are produced during inflammation, for example. And even more in immune cells specifically, such as macrophages, for example, one electrophilic metabolite called itaconate can be produced in response to pathogen infections. And this is because it is derived from the Krebs cycle intermediate, cis-aconitate, during the transcriptional upregulation of an enzyme cis-aconitate decarboxylase, which produces itaconate. Itaconate, in comparison to the lipid-derived electrophiles, for example, such as for hydroxynonenal, is much, much weaker as an electrophile. And even compared to most exogenous electrophiles, it's really very weak. But it accumulates to millimolar concentrations. In activated macrophages, both human and mouse, itaconate is probably the most abundant metabolite when these cells are stimulated with a pro-inflammatory stimulus. And the consequent activation of NRF2 then plays a crucial role in the resolution of infection. For example, in mouse models of sepsis, NRF2 knockout mice do not survive, unlike wild-type mice, because they lack this ability to adapt and counteract the stress that is imposed by inflammation. This probably cytokine storm that cannot be overcome in the absence of this role for resolution of inflammation. Another example is the Krebs cycle metabolite fumarate, which can also induce NRF2 by binding to cysteines in KEAP1. And it's interesting that there is a quite recent study showing that NAD increases the biosynthesis of fumarate, which then activates NRF2, and that in turn blunts inflammation. And finally, there is also another metabolite, which is a derivative of tryptophan and derivative of kynurenine metabolite of tryptophan, which is electrophilic form of kynurenine called kynurenine carboxyketone, which also has been shown recently to activate NRF2 and promote the resolution of inflammation. And maybe just to mention one more metabolite, methylglyoxal, because it is important that it accumulates, for example, during diabetes, also in cardiovascular disease and some neurodegenerative diseases. And methylglyoxal, and that is one example how metabolism is linked to stress response. Methylglyoxal is a product of a glycolysis, byproduct, which then activates NRF2. And NRF2, methylglyoxal can be damaging, but the activation of NRF2 increases the transcription of glyoxylase 1, which is an enzyme that metabolizes methylglyoxal, bringing the homeostatic state of the cell.
What you're telling me is makes me think about also energy production, because fumarate is part of Krebs. And we know that the full functional Krebs cycle is very important for producing FADH2, NADH, and therefore, the good functioning of the respiratory chain, mitochondrial respiratory chain. So when you were talking and explaining about this, I was thinking, if fumarate is accumulated and the Krebs cycle is not continuing to function as we normally expect, I imagine that here there is also an effect on energy levels in the cells, which again is probably kind of regulation in relation maybe to infection. We know that mitochondrial function usually during LPS activated macrophages is decreased, whereas glycolysis is increased. And I am sure that these are, again, complex mechanisms, but there must be some, again, connection. Disruption or alterations in important metabolic pathways that are there for energy production, but they are also there for signaling. And at the same time, we may have, again, a consequence for energy production in the short term and maybe in acute conditions, which are exactly the conditions that we find when NRF2 is activated. Very interesting.
Yes. Yeah, exactly. Probably that's why we also find effects on NRF2 for mitochondrial function, it is important. We don't know exactly why, but it is definitely supporting mitochondrial function and adaptation to stress.
And adaptation. Yes, this is the key word, adaptation. So the plant-derived compounds, again, going back to our exogenous and endogenous metabolites that are important for NRF2 activation. So the plant-derived compounds that induce NRF2 are often molecules that are synthesized by plants during stress conditions. Again, infection of plants, lack of water, so that these stress signaling molecules in plants become effective activators of the stress response in mammalian systems. And I find this really, really fascinating, this connection between plants, metabolism, and affecting mammalian cells' metabolism and stress response. Can you give us examples of some of these molecules? And maybe you can also tell us how you see this plant-animal interaction in the context of evolution and adaptation to the environment, not just what happens inside us, but what we are surrounded by.
Yes, sure. So one example of a plant-derived compound that activates NRF2 is the isothiocyanate sulforaphane. We're exposed to it when we consume cruciferous vegetables, for example, broccoli. In fact, sulforaphane is the most potent naturally occurring NRF2 activator that we know. What is particularly interesting in terms of evolution and environmental adaptation is the fact that the plant produces a precursor of sulforaphane called glucoraphanin. Glucoraphanin is biologically completely inert. However, the same plant makes an enzyme called myrosinase, which resides in specialized cells, and it is not in contact with its glucosinolate substrate. So it's only when the plant is injured, for example, when we chew it or when an insect bites it, then the enzyme and substrate get in contact because the tissue integrity of the plant is compromised. And this leads to the formation of sulforaphane and also other degradation products. And it's interesting that some insects, for example, which we call specialists, have developed special proteins in their gut, allowing them to divert this myrosinase reaction away from the formation of the isothiocyanates. And we think that it is very likely that in parallel to acting as deterrent, presumably, to the insect, these isothiocyanates may help the plant tissue to heal by combating oxidative stress, because plants also have quinone reductase. They have glutathione or similar molecules. So they need to heal their wounds when they are wounded. So I think it's fascinating that some of these properties overlap between plants and animals. And it is this ability probably to isothiocyanates. Nature is very economic, so it uses the same molecule for different purposes. On one side, maybe to deter the insects, because some of sulforaphane is not particularly pungent, but some other isothiocyanates, such as allyl, for example, or you've experienced, most of you, wasabi. It's really pungent, so that definitely the plant uses as deterrent for insects. But they also use it for their own healing processes, and when we consume it, they'll help us heal as well.
Interesting, very interesting. So just a little question in addition to this. So plants, do they have their own NRF2 master regulator?
Oh, that's a very good question, and I don't think we know that. We do know that they produce some electrophilic molecules, such as malondialdehyde, for example, which activates very similar transcriptional program. NQ1 is activated, some of the enzymes that are like glutathione transferases, but we don't know what is the transcription factor that mediates it. So it is very likely that there is something like NRF2 that does it, but it's not known, to my knowledge, anyway.
Yes. Well, if you don't know, I don't know who else could know. So in relation to these plant-derived NRF2 activators, you have mentioned the ones present in broccoli, and this is a very well-known information. We hope that people are encouraged to eat broccoli because of these protective properties of the isothiocyanates that they contain. What are other plants that contain important levels of NRF2 activators that you think we could include in the diet to increase our resistance to stress?
Yeah, there are many plant-derived phytochemicals that have been shown to activate NRF2, and to various extents. Some of them probably beta activators than others. I already mentioned that sulforaphane is the most potent naturally occurring NRF2 activator that we know. But there are many others. For example, resveratrol, which is produced in the skin of red grapes during fungal infection. It's known as phytoalexin because it's produced under this specific condition, is one example. We don't know exactly how resveratrol activates NRF2, but it definitely has beneficial properties in terms of and anti-inflammatory effects. It's also present in red wine, but I must say the amounts are so tiny that I don't think that we can get enough just by drinking wine. We have to go to the plant, to the fruit.
Yes, it's funny that you say that because I used to write when I was in the UK. I used to write for a little journal that was distributed to the Italian community where we were living around London. And the editor of the journal used to ask me to write articles about nutrition. And one article I wrote, I remember, was on resveratrol. And I made a calculation of how much red wine we should drink in order to achieve the same amount of resveratrol that was used in studies. And I remember I calculated they were 332 glasses of red wine that we needed to drink in order to reproduce the concentrations that were found were used in the studies.
Yeah.
So again, as you say, there is a little bit of difference between drinking wine and expecting that the resveratrol in wine is going to give us this protection. But there is a point that I think is important. When you were saying that resveratrol and other naturally derived compounds are present in different foods, obviously, and they all have different potency of activating, depending also on the concentration that is present in the food. I was wondering again if there isn't a similarity between the different activators that you talked about before that affect specific cysteines and therefore may have also different effect on the transcriptional regulation downstream, and whether also this fact that there are different potential NRF2 activators present in different foods, but of course in our diet, we can combine all these different foods. We can eat grapes, we can eat broccoli, we can eat, tomatoes and other things. Curcumin, we were talking offline about curcumin. We can eat these foods in a meal, a combination of NRF2 inducers that maybe, will work together and will synergize to induce. Do you agree with this concept? Do you agree with this possibility?
Yeah. completely agree that there might be synergism. And also, they may not activate NRF2 in the exactly same way. You already mentioned potentially they could affect different cysteines of KEAP1, but also, now that you mentioned curcumin, curcumin has been also shown to activate NRF2 by inhibiting the interaction of KEAP1 and P62. This is another way of upregulating NRF2.
Yes.
So it is possible that if NRF2 is activated by different mechanisms, there might be some synergies in the overall effect. Again, even if the downstream target genes are the same, the duration of activation would be different if the mechanism of activation is different.
Yes, so in a way we are coming back to the idea that it's very important to have this varied diet. And if we think about the biomolecular mechanisms that could justify this approach of eating well, there are actually real sound biochemical explanations for this kind of attitude in nutrition. Very interesting.
Yeah, definitely. One thing that I should point out, though, is that, of course, plant-derived phytochemicals have multiple cellular targets. So when we speak about KEAP1 and NRF2, these are by no means, they are only targets.
Absolutely.
Again, nature has evolved to be very economical and utilize the same molecule for multiple purposes. So curcumin specifically has also a very strong anti-inflammatory effects that are partly mediated by NRF2, but partly not, through direct inhibition of NF-kappa B, for example. They do affect other signaling pathways, and ultimately that might be contributing also to their beneficial effects.
Yes.
So we definitely need to eat a variety incorporate variety in our diets for maximally beneficial effects.
Yes, and because NRF2, of course, I'm happy you're pointing out, we strongly believe in NRF2, but it's not the only important biochemical mechanism that we have in our cells. You have worked for a long time and very productively in the laboratory of Dr. Paul Talalay. We have already mentioned him at the beginning of our interview. And he was a pioneer in the field of cancer chemoprevention, who sadly he passed away a few years ago. I'm just curious. I want to know if you can tell us how was your experience of working with him in his lab, and what are the major lessons that you learned from him as a mentor?
Oh, I had a wonderful experience. I was really very fortunate to be able to work with him because Paul Talalay was a remarkable person, so charismatic, very inspirational. He's my scientific father and my hero, really. So I really miss him. But he did have a full life, a wonderful life. And I've learned a lot of things from him. So I can spend the next hour talking about them. But maybe if I can just say 2 or 3 things. What I learned from him is that, and I'm trying to convey that to my students as well, is that you can never be too careful with your experiments. Every detail really matters. And every detail should be carefully recorded. And then if you have made an observation or you have a result that is even the opposite of what you expected, you shouldn't ignore that, but you should view that as an opportunity to make a discovery. Because most discoveries come from the unanticipated results or the odd observation on the side of another main question that's being asked. And on this photo of Paul Talalay, I hope the viewers can see him, first of all, holding the molecule of sulforaphane, the space-filling model, which was his favorite molecule by far, and also the inscription on the wall behind him. This inscription, I'm told, I don't know Chinese, could be translated, and that's very American style of translation. Discovery is our business. And he actually inherited that from his mentor, Charles Huggins, at the University of Chicago, who is also a Nobel laureate for the treatment of prostate cancer. And so, yes, discovery is really what our duty, perhaps, as researchers, as human beings, and helping others. Another thing that I have probably learned is that we often get overloaded, and we have to really think carefully and prioritize. He used to say, if something is not worth doing, it's probably not worth doing it well. So we should really keep our efforts and focus them where we think we have an opportunity to make progress and make a difference. So these are some of the things that I've learned from him, and I hope they will be continued by my students as well.
It's very sweet to hear what you are saying about Paul Talalay and your experience. And I think there is so much wisdom in what you are saying and what he was teaching you, and I'm sure the rest of the people in his lab. And it is true that we are overwhelmed now about many things. And perhaps we have this perfectionist attitude that everything we do, although it may not be so important, we want to do it well. I think there is a positive thing, it's also a positive attitude, but maybe we should also try to prioritize and say no to some of the things that are not so important for us. But, it's really very interesting but was he in the lab regularly? Was he? Yes, he was.
Yes.
Supportive, yes.
Yeah, he was in the lab. He was very supportive. And sometimes he really, if we're introducing something new, a new technique, he always wanted to see and even try for himself. And other times, especially when it comes to working with animals, we needed him on some occasions to come. And I remember once we were tagging the animals with ear tags with numbers for identification. And one mouse had stuck her hind limb into that earring. Oh. And the poor thing was really not well. So, but yet she didn't let us disentangle this. It was her nail that was very long. So it was only when Dr. Talalay came and held the mouse, she just calmed down completely.
He had this power of some quiet energy, and even the mice thought, now everything is fine. I can relax. Oh, excellent. So interesting. Well, I think the messages that you said were transmitted to you by Paul are definitely something very important for the younger generation of scientists. There are many other things that we and they have to learn, but these definitely are, fundamental messages. I think we are getting to the end of this conversation, which was really, really interesting for me. And I knew you would give us wonderful insights, but as usual with my conversations, I get even more than what I'm hoping for from these interviews. So I need to thank you, Albena, for your time and for your excellent explanations. I really need to thank you, and I hope the listeners will have the same enjoyable time as I did listening to the podcast. And thank you again, and I'll see you the next time.
Thank you very much, Roberta. It was a pleasure and an honor to get this opportunity. Thank you.
Thank you, Albena.